Good afternoon and welcome to this very special occasion. For those of you who don't know me, I am Frank LaFurla, the Dr. Lionel and Faye Eng Dean, and I'm truly honored to celebrate the 60th anniversary of our wonderful school. Before we begin, okay, one moment. Before we begin, I want to take a moment to acknowledge someone whose extraordinary generosity has transformed our school. His vision and philanthropy have redefined what is possible in the life sciences at UC Irvine. And his historic gift not only gave us our new name, the Charlie Dunlop School of Biological Sciences, but also set us on a path towards a more ambitious and impactful future. So, Charlie, I am particularly grateful to your belief in our mission and in the power of science to improve the lives of everyone in our society. And so I will ask everyone in the audience to give us a very robust anteater a round of applause to Charlie, who's sitting here. So I also have to start off by expressing my deep gratitude to the wonderful staff in the dean's office. Their dedication, creativity, and hard work make everything possible, including this. And I have to begin by acknowledging Benedict Shipley, who's right here. She is my chief strategist, trusted advisor, and yes, even my occasional therapist. And I really couldn't do this job without her. So thank you, Benedict, for always being there for me. And also have to give an acknowledgement to a few other folks in the dean's office: Lisa, Hazel, Madison, Ethan, Lauren, Randy, Naveen, Emily, Ritwick, and Dowie, also for their tireless efforts. And while we're at it, let's give a round of applause to all the wonderful, incredible staff in the school, because they are truly the unsung heroes that keep us running and lift us up and make our community strong. And every one of them plays an essential role. You'll learn a little bit more about this as the program progresses, but for those of you who don't know, our school holds a very unique place in UCI's history. We are one of UCI's original founding schools, alongside with engineering, fine arts, humanities, physical sciences, social sciences, and the Grad School of Administration, which later became the Mirage School of Business. So from the very beginning, we have helped establish UCI's enduring tradition of discovery and innovation. And as you'll hear, and I'm sure one of our speakers, Professor McGaw, will tell you that the very first student to enroll at UCI was in the School of Biological Sciences. A very fitting beginning for our school. Even more impressive, over the last 6 decades, we have conferred approximately 40,000 degrees in the life sciences, shaping generation of scientists, researchers, and clinicians, and leaders who have gone on to make a significant contribution to our world. So that is really quite impressive. It's hard for me to believe that I joined this campus over 30 years ago and that I've been a part of the campus history for about half of its existence. My own family story is pretty intertwined at UCI, as we moved here shortly before my daughter Monica was born, and Monica is right here. So Monica, give a wave out to everyone. And so, yet I know there are a lot of faculty, alumni, and staff who arrived even before I did, and whose contributions have had a lasting impact and who share the same deep connection to this remarkable university as I do. So as we celebrate the 60th anniversary of our school, I can't help but feel a tremendous sense of gratitude and admiration for what we built together. 60 years ago, a bold vision took place here, a vision that biology could transform the world, and it certainly has. I think you will hear later that we didn't get here by copying our way to the top. We got here by breaking new ground, and I'm sure we'll hear more examples of that spirit throughout today's program. But celebrating our history is not just about taking a victory lap, it is also about deciding what comes next and having the courage to shape our future. Change has always driven success, and we must continue to meet the opportunities and challenges ahead. And I always say if anyone should understand evolution, it should be biologists. And towards that end, we are reimagining our school and how it can be at the cutting edge of modern biology. And I am personally thrilled to announce that we will soon welcome 2 new departments to our community. Systems biology, which will be led by Dr. Arthur Lander. Arthur's here in the front row, that's the third row. And integrative and comparative physiology to be led by Dr. Matt McHenry. And Matt's here somewhere over there towards the back. So we should give them a round of applause for taking this on. So these additions underscore our commitment to remaining at the forefront of discovery and education to prepare our students for a world that demands both depth and integration across the life sciences. And as we celebrate our success, we must be mindful of the enormous pressures facing higher education today. Now more than ever, society needs research universities and schools like the Dunlop School of Biological Sciences. They need us to be active voices for truth and reason, to help dispel myths about science and medicine, and to champion evidence-based thinking. I believe very strongly that we have a responsibility not just to discover knowledge, but to communicate it, to educate and inspire others, and to engage with the public in meaningful ways. We also have an obligation to hear perspectives from all sides of the political spectrum, not just the ones we prefer, because true understanding and engagement begins with dialogue. Yet I firmly believe that research universities remain among this country's greatest strengths. And the University of California in particular stands as a beacon, the pride and joy of California, and truly the envy of the world. Every day, we prove that when you invest in education, research, and human potential, society moves forward. So as we honor 6 decades of excellence, let us renew our commitment to the next 60 years, to discovery, to innovation, to service, and the belief that science can make life better for all. That, to me, is the true spirit of this celebration. Our story is, of course, still being written, and today's program reflects that continuing journey. So following my remarks, we'll have the privilege of hearing from 2 remarkable individuals whose impact on this campus cannot be overstated. Chancellor Howard Gilman and Distinguished Emeritus Professor James McGaugh. Chancellor Gilman has been a transformative leader, guiding UC Irvine through a period of tremendous growth and achievement in the modern era. And from Professor McGaugh, we will gain a precious perspective, that of someone who is not only here at UCI's foundation, but who also quite literally helped build this campus from the ground up. I think hearing from him today is truly a rare and meaningful opportunity, a chance to listen to someone who both witnessed and shaped the earliest days of our university. Afterwards, we'll have the pleasure of hearing from 4 distinguished faculty members, each representing one of our 4 departments. Arthur Lander from the Department of Cell Biology, Developmental and Cell Biology, Jen Martini from Ecology and Evolutionary Biology, Andrea Tenner from Molecular Biology and Biochemistry, and Diego Pizzagalli, our new hire in Neurobiology and Behavior. So together, we share the excitement and promise of their respective fields. They will offer us a glimpse into the groundbreaking research and discoveries that take place here every single day. So it's my sincere hope that the next 60 years be defined by the same curiosity, courage, and conviction that built the first. And with that, it's my great pleasure to introduce UCI's 6th Chancellor, Howard Gilman. Thank you, Power.
Thank you, Frank. Good afternoon, everyone. God, what a pleasure it is to be here. What an amazing opportunity to celebrate one of the great accomplishments in higher education and a cornerstone of UCI. By the way, I arrived here 12, 13 years ago. One of the first decisions I had to make was who should be the Dean of Biological Sciences. I tripped, luckily, into the decision to say that Frank should be that person. I hope it was a good decision. He's done a tremendous job. Those remarks are great, Frank, so hats off to Frank. And hats off to me for making that decision. It is such a pleasure and a true honor to be here for this occasion. I mean, congratulations to 60 years, an unbelievable 60 years. You can take for granted universities exist. We're part of universities. We do some nice things. But when you think about what it took to create this university, this school, what as a result of that we have been able to do to transform knowledge, to create new vistas for discovery, for societal impact, and the tremendous impact that we've had on generations after generations of students. It is breathtaking. There are few such institutions in the world that can aspire to have such an impact year after year after year. And so it is completely appropriate and awe-inspiring for us to take a deep breath and think back over 60 years. You don't just celebrate it's been 60 years. 60 years is just a number. You just have to be around long enough to celebrate a 60th anniversary. What's truly awe-inspiring is what we've been able to accomplish and remain committed to even today. All of those who have taught and researched and worked at the school, the tens of thousands of students who may otherwise have never had an opportunity to enter this world of inquiry and discovery and find a passion and then make a commitment in their lives to some of this world. So many of you have contributed, but you are part of a legacy of people who decade after decade after decade have contributed. And I hope that part of what you're feeling is not only your pride in being part of an institution that has such a great impact, but being part of a legacy of people who came before you that created the opportunities for us to have the impact that we have today. So hats off to all of you who are still doing the work. And this is an occasion for us to reflect on the people who came before us, where we have the privilege of making a contribution because they did such a good job before we arrived. And as you know, from the moment that this campus was created, our commitment was not simply to be another campus like every other, but to be a place where the best minds could come to do here what was not possible for them to do in their established institutions. That's the great opportunity of a young new campus, right? It is not, as Frank said, to copy your way to the top. It's to bring people together and say, look, at Harvard, at Yale, at Princeton, the existing structures have been very successful for those institutions. There is no incentive for us to think in a free way about how you would do things differently and better. But if you have a blank landscape, and maybe we'll hear more about it, not just a blank landscape, nothing but barren ranch land. If you were to come here and look across this ranch land and imagine what it could be like if, unlike Harvard or even Oxford or Cambridge for that matter, you could think fresh. How do you want to organize this activity? The best minds. That's what attracts the best minds. My goodness, what could we do here that we couldn't do otherwise? And of all of those founding schools that were here, did any of them do a better job than biological sciences of inviting faculty who understood at the deepest level what it would be like to take advantage of that opportunity? Of course not. You know, in the 60s, you all know this better than I do. I'm just a political scientist, but I'm also a historian. I read books. I know the history a little bit. You know, plant and animal biology kept apart by long exacting structures. I'm surprised they didn't have departments of zebras, departments of chickens, departments of carrots. No, but there was a transformation that was happening, sparked by the deciphering of DNA, the connections among the various disciplines. They were all becoming much more apparent. So this nascent school was created with 4 departments of a sort that could not exist in the fancy places that were so full of their accomplishments and their prestige. And we showed them, right? Because what you want is not to copy your way to the top. What you want is to do things they can't do, and then they decide that they have to copy you because you are path breakers. Cell and molecular biology, organismic biology, population environmental biology, the first of its kind department of psychobiology. And that not only established the trajectory of the school, the culture of the school, which still manifests itself in, but what's next? Are we properly organized for the next 30 years? But it established the culture for the campus. It established the culture for the campus. And anything that we have done that is noteworthy is because you all showed us what it would be like to think outside the box with the brightest minds paving the way for the future. The very first UC Irvine course catalog contained this statement: the impact of the biological sciences upon human affairs during the coming years is certain to be very great. In its own way, a bit of a modest, it's certainly to be very great, right? But no truer words were ever written. The school has shown that with its unbelievable, groundbreaking research. It advances our mission across brain health and human disease, climate and biodiversity, data-driven biology. If you care about the future of humanity in the world, what else would you want to study? The school's organizing framework, mind, body, world, captures that idea. A healthy mind depends on a healthy body. A healthy body, in turn, depends on a healthy world. And not just healthy, an ecosystem where free human beings in a democratic society can thrive. And if you care about all of those elements, you also fundamentally have to care about the fundamental equality of all human beings, the prescience of our natural order, what all has to happen for people to thrive and reach their full potential. And you are at the foundation of it. It inspires me. So thank you for developing those themes and reasserting those fundamental connections over and over. Just to mention a few highlights: the Institute for Memory Impairments and Neurological Disorders, UCI Mind, internationally recognized; the Noel Drury MD Institute for Translational Depression Discoveries, propelling us to the forefront of something that is so much affecting new generations, as well as long-standing issues. The Center for Integrative Movement Sciences, the hub of a national network of more than 20 universities, advancing the understanding of muscular control of movement across organizational scales. There's just too much to mention. And we're noticing, and we're proud and grateful. We're proud and grateful. A few moments ago, I mentioned the visionaries who created this university, but visionaries don't just exist in the past. We have visionaries with us today, people of goodwill who recognize the value to humankind of the work that you do, not just the research, the frontiers of knowledge, but the work you do for the best student body in the history of higher education. And so on behalf of this university, I echo Dean LaFurla's thanks to our good friend Charlie for your support of the school, Charlie. It's been an honor and privilege to have your name alongside ours here at the school. And I hope that this community takes inspiration from the faith and confidence you have in it. And I hope that their work continues to make you proud of being that, of having that affiliation. I think we should hear it for Charlie one more time. And then, speaking of privilege, really, Charlie, you did a good job. It's very inspirational. And then there are the visionaries that helped get us going. A few moments ago, I mentioned the Department of Psychobiology, the first academic department of its kind in the nation. The founding chair of that department was a brilliant young professor named James McGaugh, who joined UC Irvine in 1964. You do realize the campus wasn't founded until 1965. So I don't know what the Alexander Hamilton of UCI. I mean, there's a lot of metaphors you can use. And he never left. The records are hazy, but I believe that Jim at 33 was the youngest department chair when the campus opened its doors. I mean, how lucky also do you have to get for these people? Jim is an internationally renowned expert in the process of memory consolidation, recognized with high honors of election to the National Academy of Sciences, the American Academy of Arts and Sciences, among many other well-deserved honors. 1982, founded the Center for Neurobiology of Learning and Memory and was its director from 82 to 2004. He also served as the Dean, Biological Sciences, and the Vice Chancellor and Executive Vice Chancellor of the university until he got smart and realized, what are you doing in central administration when you can actually be doing something useful in the world? So good for him. And if you ever wondered why a building is named after him, this is a few of the reasons. Jim was here at the beginning. Not only important for you, but important for all of us at UCI, that it was people like Jim who were here. So we could not be happier that he's here now. So please welcome Distinguished Professor Emeritus James L. McGaugh.
Well, first, I'd like to thank the Dean for inviting me to talk. And secondly, I'd like to thank you. Secondly, I'd like to thank the Chancellor for giving my presentation. I have a few things I'd like to add. In the late 1950s, the president of the university, Clark Kerr, and the regents decided there was time to build 3 new campuses of the University of California de novo. Now, the only campuses that were built de novo were the Berkeley campus and the Riverside campus, but the Riverside campus was supposed to be a 4-year college, sort of the Swarthmore of the West. The other campuses were all upgraded from state colleges. UCLA was San Jose State College Teachers College that UCLA was a part of. Santa Barbara was a state teachers college that was elevated to the university. But the regent said it's time to have brand new ones, and so they selected places to have them, the cities of Santa Cruz, San Diego, and the non-city of Irvine. So they looked around, and Orange County, selection was Orange County, and they said, where will we have this? Before they decided on the ranch, they decided to have it in Corona del Mar, up where the cemetery is in Corona del Mar. Except the only problem was that it was a cemetery. Now, it had no bodies, and so when the owner of the cemetery found out, he quickly rushed to Los Angeles, dug up a body, brought it back to Corona del Mar and buried it, and told the regents that it was unavailable because it was now a cemetery. So the second choice was Irvine Ranch. Now you can imagine, just an aside, what it would have been like to have Irvine and Corona del Mar. Where in the hell would you go on a road to get there and get out of there? I mean, that would have been horrible. But the view would have been nice, but the rest would have been terrible. So then they negotiated with the Irvine Ranch Company at that time for a long time. And finally, they first agreed to get the 1,000 acres for the enormous sum of $1. 1,000 acres. I think that's pretty good these days. It'd be very nice. And then they got, in 1964, just 4 years later, they got the inclusion area of 500 acres for a little over $3 million. 500 acres for $3 million. I'd say that would be a bargain too today if you could get it. Well, that was the beginning of it. The founding chancellor was Daniel G. Aldrich from Berkeley and Davis. And he invited Edward Steinhaus to come and be the founding dean of the Division of Biological Sciences. He knew Ed because Ed was from Berkeley and Dan was from Berkeley. And so they knew each other well. And Ed decided that he would come down here. By the way, I wish we had a whole body picture because he looked very much like Mr. Rogers of TV fame. And he also acted very much like Mr. Rogers of TV fame. He's a very nice man. He was very sweet. He was very pleasant, but he was also very tough. Now, as I said, he was invited to become the head of the Division of Biological Sciences. There was to have been a School of Liberal Arts, and the Biological Sciences was to be one of the departments or divisions within that. But the vice chancellor left to become the chancellor at Riverside, leaving that position open. The deans rushed to the chancellor and said, eliminate that position and make us all deans of schools, and we'll eliminate a layer. And that's how we became a school rather than a division. And it was really much to the campus's benefit, and certainly the benefit to the School of Biological Sciences to have the independence to grow under that nomenclature. Well, in January of 1964, actually the very same time that the 500 acres were purchased, I got a call from Edward Steinhaus. I was in the University of Oregon, where I had just moved just a year and a half before. I was all settled. We bought a new house and we're all completely ensconced. I had no plans to move at all. He called me and said, I'd like to talk with you about the new campus of the university and whether you'd like to join us. And we'd like to do something in your area of interest. So I thought, well, I will fly down and talk with him. And I came down and he met me at the hotel over on the corner of Jamboree and Coast Highway. The hotel is still there, but what is not there is a heliport. Why the heliport? Because there was no 5, there was no 405. The only way to get here would be Highway 1. And so the best way to get here for all of us who were recruited at that time was to come by helicopter. So I came there, he met me, took me over to the corner of Bison and Jamboree. You know why it was called Bison. There was a bison ranch there. There's a bison ranch, there were bison. And that was also the headquarters where they were designing the campus. And he took me over the vista and he said, if you look over there, you will see where the campus is and will be developed and will grow. And I was looking in the wrong direction. There was nothing to see. There was nothing to see. So he put me in his car and we drove down to what is University, which was just a 2-lane road, turned right. We went up a rutted ranch road. He got out and opened a barbed wire fence, and we drove up what is now California and around so that we could see some buildings. Oops, there we Oh, you're fighting with me here. There is an aerial view, a little more developed at a time that I first saw it. You can see the library, you can see social sciences, and you can see up at the top a building which is under construction, which was natural sciences at that time, because all of the sciences were to be in the building. And that's what I saw, except I was on the ground level, so I could only see part of that. I could see there was some rubble coming out of the ground, and that was to be the University of California at Irvine. I was not impressed. But I was impressed with what Steinhaus said, because he said, he was an invertebrate biologist. He said, you have to have a new view of biological sciences. In place of the departments that we have at the present time, which is different kind of animals and different kinds of plants and so on, what we need is an integration of levels. And so I'm going to create a division, which became a school, of levels analysis, and I want you to create the department of what he called psychobiology. And now I was a biological psychologist, but I had never heard that term before. I had never heard the term because he invented it. He made it up. He said, that's what it's going to be. Now, I went back to Eugene, Oregon, and thought it over. He didn't offer it to me. He said, we're going to discuss it. And the next day I got a telephone call and said, it's yours. So then I had to decide, and I decided rather quickly that the vision was too good to pass up. The name was too good to pass up. It was the University of California, which is the greatest public university in the United States. My PhD was from Berkeley, and the idea of coming to a campus and helping build a university which is better than Berkeley was something, yes, which I thought that would be something that I'd like to participate in. And so we moved and came here. And we moved to Newport Beach because there was no Irvine. There were no houses in Irvine. There were no streets in Irvine. So we bought a house in Newport Beach and lived there ever after. Now, after I came, Steinhaus said, I have bad news. The Dean of Social Sciences went to Chancellor Aldrich and said that he wants to move your department to the School of Social Sciences. Now, I had lived with social sciences and was happy to be out from under the influence of social science, and I didn't know what to do. So he said, I want you to go meet with the folks. The dean had an idea of hiring 4 real powerhouse people in psychology to come, but their condition was that my to-be department had to be moved to social sciences. So I met with these people and I came back. I had to write a report, and I wrote a report for Steinhaus. He went to the chancellor and he said, fine, you can transfer the department. I am moving back to Berkeley. I haven't sold my house there, so it'll be an easy move. Goodbye. And the department was not moved to social sciences. But it could have. That's an awful time. All right, so I came and I moved into my office, which was in a metal building, a temporary building which still exists over on Jamboree, on the corner of Jamboree and Campus. Campus did not exist. There was no road on campus. It was just go down Jamboree and turn left on University, which was just a little 2-lane road. All of the offices and the small number of laboratories were over there. And we could watch the buildings emerging from the campus little by little by little. So in the fall of 1964, by that time, 14 faculty members in total had been recruited. We moved into the beautiful natural science building with all of the sciences. Steinhaus, for the first time, was able to see the laboratories, and they were horrible. They had benches, but each of them had one little place in the corner where there was water and a sink, every laboratory. It was designed by a very old-fashioned chemist who thought that biologists put needles in the wings of insects, and that's what biology was. So for the first year, we lived in this natural science building being remodeled brand new, but had to be remodeled before we could do any serious work in it. All right, at the time there were 12 faculty in the school in the 4 departments, and everything was as exciting as you can imagine. It was really like summer camp, trying to figure out who we were and what we were going to do, all of recruiting of graduate students, getting our grants. As was mentioned, the very first student at UCI was a biology student, was in neurobiology and behavior. There was no registrar, so we had to register him at UCLA so he could get credit, but he was the very first student. Eventually, he went on and became a faculty member in Colorado. All right. After a few years, Ed Steinhaus said he was a little bit tired and would like to have a sabbatical leave and step away. And we thought he had earned it because he had worked very hard pulling this all together. So everybody agreed, and the chancellor agreed, and he took a sabbatical, and I was named acting dean, which is kind of interesting because my background was not in traditional biology, but Ed Steinhaus thought that it was sufficient to do the job, so that I was named it. And about 3 years later, Ed passed away, very, very sadly. And there we were. We had this beautiful new school, and we had lost our leader. So I maintained that position for several years. I became the Dean of Biological Sciences, and then we recruited for a new dean, and were very lucky to get Howard Schneiderman, who came for the third Dean of Biological Science, an outstanding scientist and an outstanding leader, just a wonderful person. And he began to take the campus, the school, even to greater heights than it had been before. So that was a wonderful thing that happened. Now, something about the faculty. We had 17 faculty to begin with, but they were all really very good. By the time that Howard Schneiderman arrived, there were 5 people in the school who had been elected to the who had been or would be elected to the National Academy of Sciences, and there was 1 Nobel Prize winner. Lee Hartwell won the Nobel Prize. He was here for several years before he was recruited away to the University of Washington, but we found him first. And he did his pioneering work right here. So that's not bad for beginning. 5 National Academy of Sciences and 1 Nobel Prize winner in an initial group of 17. That's pretty good. All right. There's much to be said about the school and its early foundation, but there's not enough that can be said about Edward Steinhaus, who had the dream, and he had the ability, he had the insight to develop the school, which was different from anything that had ever been done, and to recruit what I believe was an outstanding faculty to get it started, and we owe a great deal to him for the founding of this school. Now, if we go back to the previous slide, his building is up there, not to his liking yet, of course, but in the upper right-hand corner, you can see a building that is underground. That was the Natural Sciences Building, which ultimately was named Steinhaus Hall, and we were all pleased that we were able to get that named in his honor. I think that Edward Steinhaus would be very satisfied, and I think he would be very proud of his development and his contributions, and I am too. Thank you.
Thank you very much, Jim. That was fantastic, and I learn something new every time I hear you speak. So let's give Jim another round of applause. Okay, so we are now going to hear from four of our distinguished faculty who will give you a little taste of the excitement of their own research areas. And first up is Dr. Arthur Lander from the Department of Developmental and Cell Biology. So, Arthur.
Thanks. So I've been asked to give a micro talk about systems biology, which is a pretty difficult thing to do, but I'm going to try. So life, as we all know, is complicated. And one of the really cool things about biology is how it's complicated at so many different levels, all the way from the intricate structure of a virus to the morphologies of organisms, to the organization of ecosystems. It's incredibly beautiful and incredibly complicated. Oops, wrong way. So for 400 years, science has been trying to help us to understand all the complexity in biology. And typically, science tries to make complicated things understandable by breaking them down into parts. And in the case of biology, we've often discovered the cells, the molecules, the genes. We've connected them together in different ways. We've even been able to sort of quantitatively look at how changes at one place affect changes in another place. But the problem is when things get complicated like this, where you have lots and lots of interactions and huge big networks of things, this approach really doesn't work very well. It's very difficult to understand what's happening when you have these kinds of interconnections. Well, in biology, the real interconnections are much, much bigger than that. This represents just the genetic interactions within a single-celled organism. And this represents all of the interactions among all of the different proteins found in a human cell. So just try to take this apart piece by piece and see if you can understand it. It's not very likely to work. So, not surprisingly, if you look at the major problems of biology that still exist here in the first quarter of the 21st century, a lot of them have been around for a long time, and they're still unresolved. We still don't really know why there's cancer. We don't fully understand things like dementia or psychiatric illness. We don't know why there's such a high prevalence of birth defects, or how to understand what climate change is going to do to our ecosystems. These are all problems of that kind of complexity. They all involve very heavily networked systems, whether it's networks of cells or networks of metabolic products, or networks of species interacting with their environments. They all have that character, and that's one of the reasons why we still really haven't solved these. So interestingly, in the last century, a lot of effort was put into trying to understand these problems that involve big networks and lots of interactions, and most of those problems related to issues of human systems rather than biological systems. So economics and management and politics and finance and engineering, and there's lots and lots of books that you can download and read about general systems theory or systems thinking or thinking in systems and so on. And all of these were attempts to get at this sort of general idea of how do large network systems behave. But it really didn't take hold in biology until about the turn of the century. Right around 2000 is when suddenly biology starts jumping on this bandwagon of systems and networks. And so there were a couple of things that were necessary to bring biology to this point. And one of them is biological big data. So I remember training at a time when you got data points one at a time. Actually, it was more like one graduate student at a time, right? And the student would labor for 5-6 years and maybe measure 100 or a couple hundred things. And that would be a thesis. And that would be the way that biology progressed. And in a very short period of time, particularly over the last 25 years or so, we can now measure 100 million things at a time in an afternoon, right? And in fact, the collection of biological big data now far surpasses that of astronomical big data climate big data, many of the other areas that you hear about. Biology big data is already well beyond that and growing faster. And the second thing that's related is computational power, right? I mean, just this little laptop has something like 100,000 times more computational power than what sent people to the moon. And a supercomputer has about 1,000,000 times more computational power than that laptop. So you can add up all those zeros. It's a huge ability to handle the kinds of data that we're now gathering. And so really only in the last 25 years has it really been able to put biology in these terms of we're gathering so much information and we need to be able to process it. And you can see since about 2000, there's been an explosion of literature, of books, of meetings, of publications, all focused on this area of systems biology. But systems biology isn't just about throwing computers at big data, right? And nowadays, I think we're all aware of the tremendous power that you can have by throwing computers at big data, because we've all seen things like ChatGPT, right? We know how something can take an enormous amount of information and distill it down in ways that it can make really important predictions, like what would be the next word in a sentence, or what would be the next sentence in response to your prompt. But prediction is not the same thing as understanding, right? Prediction is limited to telling you how something is going to behave just like something always behaved in what you trained it on, right? Something new happens, prediction isn't going to help you. And unfortunately, just computing on big data really doesn't give you a sense of what's the reason for things happen. It's not very interpretable. And so systems biologists, they need to answer some very fundamental questions, like what's this biological network for, right? That's not going to emerge from just sort of computing on it. What would happen if the circumstances were changed from the data we have now? What happens when these systems break? Because essentially all of these diseases or planetary crises that can result in biology are when these network systems break. And how would the results differ if we'd left some parts out? This is an important piece of science, is that we always have to deal with the unknown unknowns. So if you just train something on what you know, you're never going to learn about the things you don't know. You're going to learn a lot of things that people knew, but you're not going to learn anything that no one ever knew. And so consequently, systems biologists have had to turn to so many other fields where complex systems have been analyzed, including mathematics and computer science, and a lot of engineering, physics, genomics, informatics. In fact, I'd have to say that from the standpoint of interdisciplinary synthesis, this is the greatest example in the history of science of bringing together input from a large number of disciplines in the service of one new one. So it's very exciting to actually be at a time when this is all coming together in the service of a common goal of making these really, really complicated systems understandable. And so thankfully, UCI was lucky enough to get in on the ground floor of systems biology. Ever since the early 2000s, we've had centers that have been developed, a graduate program since 2007, one of the oldest graduate programs in systems biology. We've focused on cancer, on cell fate. And so far, these groups have brought in over $70 million in just grants focused specifically on systems biology for large groups, or training grants, training students in systems biology. So we've had a really nice run. And as Frank mentioned, we're expecting in 2026, that is in a few months, we're expecting to be able to launch a Department of Systems Biology within the Dunlop School of Biological Sciences. This is the first new department in 60 years, so we're very proud of that. Here's the founding core faculty. for the Department of Systems Biology. These are the individuals within the school who've agreed to move all or some of their appointment into the department. And these are some of the areas that they work on. You can see everything from the theory of evolution to how cancer operates, to how to do computation within biology, to animal development, and so on. And you can see there's no one topic here. There's no one organism or one particular approach. Rather, there's an approach to all of biology by trying to find the fundamental principles of design that cut across these many different areas. As I said, systems biology draws heavily on other disciplines, and here within UCI, we have people in all of these other disciplines outside the School of Biological Sciences who are already actively collaborating with the new faculty in systems biology. And one of the most exciting things is we hope to very soon have an undergraduate program in computational and systems biology, which is something that we know that the undergraduates here are very eager to participate in. And we're very eager to develop new ways of teaching that bring them directly into this field, rather than going as we had to go by first specializing in one of the subdivisions of biology and then moving into this. So that's all I have to say, given the length of talk. But if you want to learn more about systems biology, please come and find me at the reception, and I'd be happy to tell you more. Thank you very much. And so I'd like to introduce the next speaker, who's Jennifer Martini from the Department of Ecology and Evolutionary Biology.
Good afternoon. Well, I'm really honored to be here today and represent my Department of Ecology and Evolutionary Biology. And I'd like to tell you just about one example of why ecology and evolutionary biology, despite the origins of pinning butterflies that we heard about, why we're still on the forefront of solving many of humanity's biggest problems. And my colleagues will not be surprised at all that my example has to do with microbiomes. Microbiomes are collections of bacteria, viruses, fungi, and other microbial organisms, microscopic organisms. And you've probably heard about microbiomes first in terms of the context of the human microbiome. And so in and on us, on our bodies, there are more than 10,000 different species that live here. And that diversity is actually pretty astounding. The genetic diversity, the number of different types of genes that those microbes encapsulate, is greater than 300 times the number of genes in your own genome. But human microbiomes are just one aspect. Outside of our bodies, in ecosystems, there also are microbiomes. And you hear a little less about these, but they're no less important. So in a handful of soil, the soil contains over 1 billion cells of bacteria. In a mouthful of seawater, you're swallowing over 1 billion viruses. So something to think about when Charlie goes surfing next time. Luckily, most of those viruses infect other microorganisms. And in fact, generally, microorganisms, despite everything you've been told since you were little, are not usually pathogens. Very, very few of them actually are. Let's see. And so hopefully that sets the tone. We are a microbial ecosystem, and we live in microbial ecosystems. And I will go one step further. We live in microbial ecosystems, and we evolved in microbial ecosystems. And what I mean by that is microbes, they've been around on this planet for billions of years, longer than plants and animals. All of our cells, our organs, even our brain evolved in the context of this soup of microbes around us that we can't even see. And so you really can't understand microbes and microbiomes of humans without thinking about microbiomes of the whole world. And it's this intersection that I think is a real tangible example of our school's theme of mind, body, and world, and how interconnected we all are. So you've probably also heard about how microbiomes are associated with health and disease. And this is definitely true in terms of the human microbiome. They're associated with everything from obesity, cancer, the efficacy of our vaccines, how well a pharmaceutical works, even chemotherapy. And we know this because we've been looking at the composition of microbiomes since genetic sequencing became so cheap and readily available. And that was around the time I got my PhD. Since then, we've also gone out into the environment and we can capture the composition there. And we can see that just like our bodies, our ecosystems are correlated and how they are functioning are correlated with the composition of microbiomes. So everything from plant productivity of our crops, the drought and heat resistance of those plants in the face of climate change, even soil fertility and the amount of carbon that's sequestered and stored in our soils. All of these things are correlated with the composition of microbiomes. But of course, some of you out there that might be scientists will think, okay, but this is a correlation. Is it really causation? Are microbiomes and their composition actually mediating or influencing all of these conditions? And so the last 20 years or so have been, many researchers have been working on this, trying to understand that. And of course, since we don't have sterile humans or sterile ecosystems, this has been a big challenge. And so some of the most dramatic experiments that have been done that show that microbiome composition matters are those that transplant entire microbiomes from one place to another. And you may know what's coming up if you know about this field, and that is the fecal transplant. And so we now have a USDA approved therapeutic where, especially if you are infected with Clostridium difficile and your treatment is not responding to antibiotics, your doctor can prescribe this treatment and it's extremely highly effective. Now, out in the soil, I wasn't involved in the fecal transplants, but I was involved in how do we figure this out for something like soil. And so my lab and colleagues in the ecology and evolutionary biology department, we were trying to do transplants of our own. I would argue a little less messy, but it still required a different kind of creative work. And so we needed to be able to engraft soil microbiomes onto soil material and then be able to move them into different environments. And in this way, we did this by creating small little microbial cages, I like to call them, where we can move them around, but they're not dispersing in or out of those. And so in this way, we were able to tease apart the effect of the environment versus the effect of the microbiome on something like carbon cycling. And we showed that microbiome composition does matter even in the soil. So this is exciting, but I think, but what's really exciting about finding that microbiome composition matters is that then we can turn to thinking about, well, if it matters, maybe we can start to engineer it to towards our own outcome that we would like. So can we engineer microbiomes? You will think, first of all, that you might do this with something like probiotics. And in fact, that's the example I'm going to talk to you about today, but there's many other ways we can do this. In the environment, we often talk about these as bioinoculants, but they're really the same thing. And so I've already given you examples. On the one extreme, we could try to manipulate the composition by moving a whole community around. But again, this is quite messy. It's also not very scalable. Are we going to move an entire, wheelbarrows of soil across a field, things like that? On the other extreme, you can think about just adding a single strain and asking, can we perturb and move a community in a direction that we want to? And so you can go to Trader Joe's or Albertsons and buy a probiotic. You can also order bioinoculants like mycorrhizae to put into your garden. And people are doing this sort of thing even at a large scale. We also are even genetically engineering single strains to do things, including at the bottom, it's a company that's genetically engineered nitrogen fixing bacteria, and those are being used at scale in the US today. A funny story about the top probiotic I put up there next to the drink. Don't, is a genetically engineered soil microorganism. You can buy this off of Amazon. It's called Z-Biotics, the company. And the idea there is that if you take a shot of this probiotic before you go out and have some drinks, then it should reduce your hangover in the morning. In fact, I have personally tried this one with some of you in this room who shall remain nameless, and I'm happy to tell you the outcome of our results on that experiment. Maybe later. All right, I digress. So in the middle of this, though, what I hope you see across this scale is that there's a huge range of opportunity to try to modify microbiomes somewhere between 1 strain and 10,000 strains. And that's a lot of opportunity. And luckily, this is the range and the scale at which ecologists work. Microbiomes are ecological communities. I like to think of our gut microbiome as a tropical forest. Our skin microbiome is a desert. And traditional microbiology is going at this not in an ecological approach, but one microbe at a time. And this comes from the origins of microbiology, where we're worried about one pathogen at a time. So we want to just take our sterile organism, add that microbe, identify the specific gene or genes involved, knock them out, compare them to a wild type, and then voila, we know that this microbe is important. Of course, if we're going to do this for 10,000 species, it's going to take a lot of trial and error. And the problem with being in an ecological community and why we need an ecological approach is that there are thousands of species that are interacting. And I'm very glad that I just went out after Arthur, because there is another network. This is the same type of problem we see in many biological systems. All of these dots are abundant bacteria in the soil right around a root of a citrus tree. And so if you're thinking, I'm going to add in one species here, which species do you pick? How is it going to impact all of the other species? And what's going to be the outcome of the functioning of your community? That's going to be pretty hard to predict. Ecologists also appreciate that there's a lot of spatial and temporal variability of microbiomes and of communities. And so your microbiome is different than my microbiome. And so it's no wonder that there's mixed results about probiotics, because if you take one strain and I take that same strain, it's likely we have completely different resident communities in our gut. And so it's not going to affect us the same way. There's also a lot of dispersal between ecosystems. Your microbiome, your gut microbiome, is influenced by whether you grew up on a farm. It's influenced by whether you own a dog. It's influenced by the microbiomes of people you closely interact with. And so if we just, again, focus on one system at a time, we're not going to be able to engineer it properly. So the state of the art right now in microbiome science is to take one particular problem, let's say irritable bowel disease, and go to your freezer and say, okay, I have 40 strains of gut bacteria in my freezer. Let's try these 5 in a mouse and see if it helps the symptoms of this disease I'm working on. And of course, this is important research to do. We definitely need specialists in this field. And personally, my own lab is focused on one genus of soil bacteria that lives in the Irvine Hills. And that's important work, I'm the first to point out. But at the same time, if we do this one organism at a time, or even 5 at a time, it's going to take us forever to figure out how to engineer all of these microbiomes. And so instead of thinking about one problem at a time and then moving on to the next and the next, and then, even out in the environment where we're trying to restore a hillside at Crystal Cove Park, but that's going to be different than a hillside somewhere else. Same as agricultural engineering and aquaculture engineering. So what I think ecology and evolutionary biology offer here that we wouldn't usually think about if we're just studying one microbiome at a time, is we can think about what are the fundamental principles across all of these systems, and how can we use that information to accelerate how fast we're getting to new solutions, microbiome-based solutions in the face of all of these problems. Now, luckily, ecology and evolutionary biology already have a lot of theory and approaches that we can pull from. And some of them I've just listed here. But for instance, identifying keystone taxa within food web theory. These are the taxa that have an outsized influence on other organisms in their community. And so they're the knobs potentially we want to target first as we try to modify microbiomes. It's been really a pleasure and so much fun to be working at UCI and in the School of Biological Sciences. And it's here in this school where my colleague Katrina Whiteson and I established a Center for Microbiome Research in 2017. And our whole idea was this intersection of environmental microbiomes and human microbiomes, and to really start to think about the intersection here of microbiome health and restoration. And we have grown now to 23 core faculty in our center, although we are collaborating with more than 100 labs across campus. But I would like to point out that how exciting and how fast-moving this field is. There's 23 faculty and 11 we've hired just in the last 6 years. And these are our incredible assistant professors, most of them in biosci, or at least a joint appointment with biosci, and they're winning all of these awards left and right. And so it really is an exciting place for this field to evolve. And UCI has been leading the way. And one tangible way in which we've been leading here is by helping to found a national network of microbiome centers across the country, which are also working across all these different disciplines and trying to bring this type of research all together and think about microbiomes in a different way. It's really only been the last 5 years that we've started talking about microbiome science as it's a new field. And again, working across these different disciplines, training students to think about it, and losing my train of thought. Training students to think about it. And I can't think of a better place to be than at UCI, where there's so much energy and excitement and thinking about these big problems that we can solve here today. So thank you very much. Thank you. And now I would like to introduce my colleague, Andrea Tenner, from the Department of Molecular Biology and Biochemistry. There we go. Okay. I want to close. Thank you. Okay. So thank you, Frank, for inviting me to come and talk, and thank for all of you for being here. I want to talk a little bit about evolution to some extent, but I arrived at UCI 34 years ago, so I've been here over half the lifetime of the school. And it has been absolutely great. I came as an immunologist, and immunologists are really studying how the system recognizes needs or recognizes danger and then sets out to make a solution for that, i.e. kill or get rid of the particular pathogen. And it does this throughout our body and throughout our lifetime. Now, the immune system doesn't just defend, though, against pathogens, as it turns out. And so we were looking at also the fact that it repairs tissues. It clears out dead cells or cells that are no longer functioning anymore. But when I arrived here at UCI, it was a powerhouse of neurobiology. And I got sucked into the neuroscience really quickly. And it's been great. Who knew that the immune system would have that much to do in the brain? And so with a combination of being in a place which
grew up its immunology, we were able to begin to ask now, we can think, oh, okay, what if we could guide the immune system to really recognize and destroy all the cancer cells more efficiently? How could we now prevent Alzheimer's disease before symptoms even arose? Could we prevent the loss of vision? Could we guide the immune system to repair a damaged neural circuit? All of these things are the frontier of immunology now. We're not quite there yet, but it's moving very, very quickly. And I just want to mention a few things about a few examples of where we are now. So the immune system, there's not a quiz. All right. But the immune system is really developed to two parts, adaptive and then the innate. And everybody thinks about antibodies made by B cells and maybe T cells now with cancer therapies. But the innate system is one that is there on the ground all the time. Really, their job is to identify very quickly, within minutes to hours after somebody is either infected or injured, identify the pathogen and get rid of it, have a role to get rid of it. And one example of a component of the innate system is complement. And this particular system is really a dynamic sensor system. It's almost like a security system that it can sense, it can tag, and it can help destroy emerging pathogens. But I'll tell you a few minutes later that it does actually much more than that. But one of the things that's really important is that the innate system now, we know that it helps to direct the adaptive response, making sure that it's an appropriate immune response for the particular challenge. But everything needs to be balanced. And so if we orchestrate the immune system by knowing a lot about the different parts, then sometimes we'll want to enhance the immune system and sometimes we'll want to decrease it. But then we'll be healthier for a lot longer time. And I just wanted to bring up one thing here. You might have heard in the last couple of weeks, the 2025 Nobel Prize was given for the identification and beginning characterization of regulatory T cells. And these are cells that actually control and try to dampen down the immune response under the correct situation so that you don't get autoimmunity. So, and just to know that this guy was trained in the UC system. Okay, we can go into more of that later. But nevertheless, just to give an example of how we are now able to guide the immune system, I want to mention a little example here. You might have heard of checkpoint inhibitors, but here's a T cell of the immune system and a tumor cell, bad guy. And in order for the immune system, this T cell to actually function, it has to be able to recognize this tumor cell, which you can do that, but it also needs this thing called co-stimulation. You need to have two signals. You can't just fly off the handle and start making all kinds of toxic stuff if you only have one indication. So you need a co-stimulation. However, the immune system is also wants to be quite clear about the fact that, well, we don't want to go overboard here. And so they have built in some brakes on this activation of the T cell so that it's not going to wreak havoc on the surrounding cells. So you have these inhibitory ones. But if you have an aggressive tumor, you don't want any kind of intrinsic brake on the system. You want it to be activated. So that's where James Allison and Hojo came in, and they decided, well, if we made an antibody to these inhibitory reactions so that we would prevent these interactions, we could jazz the T cell to be able to respond much more appropriately and really nail the tumor. So that was good, and that actually won the won the Nobel Prize in 2018 in medicine. So what you were doing then was really releasing the built-in brakes on this T cell function to enhance that anti-tumor immunity. But not all tumors respond to this kind of treatment. And so just an example of what is happening in molecular biology and biochemistry is that Dr. Roberto Tinoco is now looking at additional alternative therapies that will work in resistant tumors. And he's now shown that with his new antibody treatment, this anti-PSGL1, he's able to reduce the growth of tumor cells in mouse models dramatically. So there's the growth of these tumor cells, and with his treatment, you can reduce that, more than half. So that's really a great start. And really watch this space, because things are going to be happening, and what we might be seeing in the future is sort of a cocktail approach that is actually beginning to be ongoing now in the therapies, of trying different therapies and combining them so that they will enhance the immune response even more. So now I'd like to just sort of turn around and go back to complement. Okay, complement is evolutionarily ancient, before B cells and T cells were around, but also it has had all this time to evolve, and so it doesn't just kill pathogens. Indeed, we found out that the complement system, like Taylor Swift, is an influencer. And it does this stepwise sensing and signaling to deliver the appropriate immune response, given the type and the magnitude and the dimension. So it's an influencer. That's a good thing. So these are good things that the complement system can do. In addition, something that we did not know in the previous century that we only found out in the 21st century was that complement actually plays a role in the nervous system. And this is a synapse, and these are cells of the brain, and the synapse is on a neuron, okay? And these cells are looking at the synapse and saying, is this a weak synapse or is it a functional synapse? And if it's a weak one, it's going to have these complement components. Whoops, shoot. Wait a minute, complement components all over them, and then it's going to ingest it and it'll trim it. And that's really important during normal nerve development. Also for adult learning. So these are good things about the complement system. But gee, you get excessive synapse, and you will have really problems in neurodegenerative and perhaps then neuropsychiatric disorders. So there's the good things and the bad things. And finally, one more example of this is when overactivated, the complement system can really generate a lot of inflammation, which is very detrimental to tissues. And so an example of that, which I'll come back to, this is Alzheimer's disease, an amyloid plaque in the brain, and these microglia are responding to that plaque, and you can see they're red, they're really angry, right? And they're actually producing a lot of neurotoxic molecules. And that is going to be detrimental to the neurons, which are not on this, but are in the brain. And that's going to be neurotoxic and cause real problems. So, okay, so all these different things really enables you to be able to have specific targeting, but it also requires specific targeting, because you can't just blow out the whole system, because then you can get rid of a lot of the good stuff. So we applied this to Alzheimer's disease. And we just real briefly here, I'm going to point out that in an Alzheimer's disease human brain, you have these plaques, these amyloid plaques, but you also have deposits of some complement components down here, okay? In a non-demented individual, you don't see any of these complement components on the plaques, in the brain. And in fact, you can have amyloid plaques here in the non-demented, but if you don't have complement in here, you're still cognitively okay. So one of the things that we wanted to know, well, if this complement is here in the AD, is it a good thing or is it a bad thing? You got to know what the enemy is. What is causing the neurotoxicity if it's not these complement components that are induced, or is it? Do we want to knock the whole thing out? Well, we were able to see by knocking out one component of this complement receptor in mouse models of Alzheimer's disease, and we were able to demonstrate that we could actually protect against memory loss. And you can see here that it really rescues neuron dendritic processes. Here's the AD mouse model. It looks like a scarecrow here relative to a normal neuron. And if you have the AD model, you get the scarecrow, but if you don't have the C5AR1 receptor, it's just like normal, and you've protected memory. So what is causing that? What we know that this particular receptor is on microglia in the brain. That's particularly surrounding. There's a plaque there. It's not stained, but this is a mouse brain, and you can see these are microglia. But how do you get from C5AR1 on microglia to being killing of neurons, being neurotoxic? Well, in collaboration with the Ali Mortazavi's lab in development on cell biology, we were able to do this single-cell RNA-seq, and we were able to find indeed these microglia that were in the brain of an Alzheimer's disease mouse. They were making all kinds of pro-inflammatory cytokines that were toxic to neurons. But if you, But if you treated that with an antagonist, you could stop the production of genes that were inflammatory, and you could suppress excessive synaptic pruning, but you left some of these other protective responses to this danger intact. And so you're preventing the loss of behavior and neuronal integrity with this receptor inhibitor. So that means that we should really just target C5AR1 and not knock out the whole complement activity, because now you can have other forms of immunity in both the brain and the periphery. And we also know that they lack toxic effects. So the next step then, which we would like to be able to think about getting started, would be a clinical trial, because you don't know until you try it in humans. But the other thing that you'd like to be able to do is actually, we know that some of these proteins have positive effects. So if we could find out how to ramp up those positive effects, again, we can have another cocktail effect here, which I can talk to you about at the reception. And we can try and see if we can get a lot of benefit for AD patients or people in pre-AD as well. This doesn't really knock out the value of other AD therapies, but this is a general one, as it might be a general one for neurodegeneration, because they all are involved in this inflammation and all have some similar genetic signatures. So with that, I would like to just move on to mention, this is people that have done the work in my lab, but I bring this up because really science and biology are team sports. And we need to acknowledge individuals, and particularly this Sophie Chu was my lab manager for over 17 years. She's been outstanding. And then all the people at UCI, of course, that are there, lots of undergraduates that are really wonderful. And then lots of funding. This is all funded by NIH, as well as some other Alzheimer's associations, and then donor funding, which makes us be able to do much more impactful work. But individuals are really, really important, but teams and dream teams are also critical. And so from the very beginning of the small teams in the labs, as it grew, to teams that are brought together in the different departments in biological sciences to make Model AD, to international collaborations. This is Trent Woodruff, we've known for over 20 years, and he's been very instrumental in all of our work done. But the point here is biology and science are team sports, and we need to all work together. And that's what the School of Biological Sciences here at UCI does incredibly well. So I'm going to say thank you. And just to take home, everybody loves complement. So thank you very much. And I'd like to welcome Professor Diego Piazzagalli as our next presenter. Thank you. Close.
Thank you so much. It's really a treat to be here today and have 10 minutes to share with you some of our vision for this I assume, maybe the youngest institute within the School of Biological Sciences, which is a new institute focused on depression. Depression, of course, as was mentioned at the beginning, really kind of a daunting problem for our society. Around the world, around 280 million people make criteria for depression. This really boils down 1 out of 4 people will experience depression at least once in their lifetime. And a couple of years ago, depression actually became the leading cause of disability worldwide, surpassing cardiovascular disease. And it's not only associated with quite profound personal suffering, but also staggering cost to society. For example, in 2023, depression costed to the U.S. Society over $380 billion. And unfortunately, there are specific sectors in our society, especially our youth, our adolescents, where the rates of depression actually have been going up. Actually, just an increase of, doubling of the prevalence among adolescents. And there are specific kind of challenges that we face in this field. For example, we have actually quite a lot of, several antidepressant treatment, different classes of medication, psychotherapy, even neurostimulation. But it's interesting to note that, with all these different approaches to treat depression, about half of our patients will actually respond. So, this leaves half of them where basically these interventions are not beneficial. We also have been very interested, and this is something that we'll definitely emphasize quite strongly in this new institute at UCI, that there are specific individuals that are at increased risk for depression. For example, youth that happen to have a parent with depression, where statistically speaking, 30 to 40% of them will go on and develop depression themselves at some point in their life. Of course, we like to know who among them are at risk for this disease so that we might be able to intervene with prevention strategies. And similarly, although for some people depression might occur once in their life, maybe while going through a difficult situation, for the majority of our patients, individuals with depression actually is a recurring disease. That is, symptoms might occur. With appropriate treatment, they might disappear, but most people actually will have a recurrence. And again, here in this new institute, we're very interested in identifying potential markers that we could use to know who might be at risk for recurrences, because this might allow us to intervene, maybe with some prevention strategies. These kind of sobering statistic and facts are compounded by a couple of additional challenges that we have in the field. So with very few exceptions, if you think about pharmacology for depression, there's been actually, I would say, quite limited innovation. So the field has developed what we describe as Me Too drugs, meaning that they basically have effects very similar to the very first antidepressants that were discovered, over 60 years ago. These new medication actually have a better side effect profile, but fundamentally in the brain they have exactly the same mechanism as the very first antidepressant that again was discovered over 60 years ago. Similarly, in spite of progress, we don't completely understand the risk factor associated with depression, which again might stymie prevention strategies. And then something that, again, we really want to emphasize in this new institute is that typically in psychiatry, and this is very different from other field of medicine, basically treatment often follows a trial and error approach, right? So individuals might, for example, try a specific medication or maybe psychotherapy. We have to wait, 4 to 5 weeks to see whether the medication, for example, will have a beneficial effect. And again, 50% of our patients will not benefit. And then, this trial and error start again with maybe increasing the dose, maybe switching medication. And we are, of course, very much concerned about having uncontrolled symptoms and increased risk for suicide and other, tragic events. And so the type of research that we want to do at and this new institute is actually shown in this diagram here. This is actually a study that we recently completed. So imagine that you're actually recruiting, as we did in this study, a large number of patients with major depressive disorders. Assume that you treat them with a specific medication, for example, in this case, widely used selective serotonin reuptake inhibitor. And after 8 weeks, what you will find is that about 50% of them will have responded to the treatment and the rest will have not responded. What we did in this particular study is that those that responded to the SSRI actually stayed on the same medication for 8 more weeks, whereas those actually that did not respond to the first medication were switched to a different type of antidepressant medication, something that has a very different mechanism of action. And again, here too, if you follow patients long enough, you will see that about half of them will respond to drug B. And so the key question that we ask in this particular study is that if we now go back to the data, in this case, neuroimaging data that were collected at week 0, can we find actually some predictors that might allow us to say whether people responded to a drug B after failing this drug A? And what we were able to show in this particular study is that indeed, by using functional MRI in this case, we were able to find that the strength of functional connectivity, if you wish, the crosstalk between these two brain regions, which are two key hub in the so-called brain reward system, a set of regions which become simply activated when we are anticipating a potential reward or receiving something pleasurable, that the strength of the connectivity between these two regions actually allowed us to predict which patients ended up responding to drug B after failing drug A. With other words, we were able to make predictions about the response to antidepressant treatment, and in an ongoing study, we're using this type of predictors and biomarkers, in fact, to decide at the outset how patients actually should be treated. And this is the type of study that we will, emphasize in this new institute, where I believe that we're really quite uniquely equipped to tackle some of the challenges that I've described, because this is actually quite unique in the country and probably around the world, is that this particular institute at launch is actually bringing together not only the Charlie Dunlop School of Biological Sciences, but also the School of Medicine and also the Samueli School of Engineering here at UCI. And we will also work with, of course, all the other schools on campus that do relevant research. And I think that this will give us really a unique opportunity to bring together clinical, basic, and translational sciences that, again, are really needed to tackle some of the challenges that I've just described at the beginning. And so our vision for this new institute is hopefully to transform our understanding of the causes of depression, its treatment, and its prevention, while enhancing societal well-being. And basically, first and foremost, what we're going to do is again, to create a vibrant and inclusive and highly interdisciplinary community of scientists that are really working across units of analysis, are working across species, are working across discipline to, again, tackle this disease. And basically, the scientists will be engaged in a kind of three-pronged approach, with three fundamental missions. One, to fundamentally improve our understanding of this disease and its treatment, and hopefully preventing it. The second goal is really to build an ecosystem where hopefully trainees will want to be, and I'm personally very passionate about mentoring, as well as colleagues that are joining forces with us. I think that we really need to train the next generation of scientists in this area, particularly at a time, unfortunately, when science and medicine often is a bit under attack. And we'll pursue all the scholarly activity that typically scientists do, publishing papers, hopefully getting grants, and so on. But I think that, one of the key goals, of course, will be to try to change the needle, to move the needle with these particular disorders, and really try to have an impact on people that are facing these disorders. And really, this institute hopefully will make an important contribution, especially toward understanding, treating, and preventing depression. So I'm relatively new faculty here. I was 23 years in the Boston area, and I joined just this past January, and it has been a fantastic transition. I found an extraordinarily collaborative environment and very, very supportive university. So I cannot think of a better place to be here and tackle this disease. Thank you.
Okay. Well, I know it's getting late, but I hope you're as energized as I am by all the great science that's being done here. At our core, we are a type 1 research university, and I hope you got a little bit of a flavor of the wonderful discovery and innovation, as Diego said, that really can benefit all of society. We'll have a reception shortly, and all the speakers will be there, but let's give them a round of applause for just a wonderful talk, and they'll be happy to answer all your questions. But also, let's just thank all the donors, big and small, who allow our research to go forward. We really appreciate your generosity and ultimately the impact that it will have on our society. So thank you all. And now I'm just going to invite everyone to join our reception. So thank you all.